I would like to welcome all the participants to this first session of the PedCRIN webinar series. The topic today is Tools for the Management of Pediatric Trials. That the tools that will be presented today are, and in the next webinars, because this is a series of webinars, are the results of a project that is called PedCRIN. And that is a Horizon 2020 funded project. It's a 4-year project started in January 2017, coordinated by ACKNOWLEDGMENTS, and involving a consortium of 15 partners. And you have here the list of the partners involved in this project. The objective of the project was to enhance the capacity for the management of multinational pediatric and neonatal non-commercial clinical trials, and to conduct some pilot multinational neonatal and pediatric trials, so investigator-initiated within this project. The project was involved 6 work packages, with 6 work packages, and I just listed here the one that are relevant for the webinars, and one was the development of tools for the setup and the management of neonatal and pediatric tools, pediatric clinical trials. One was dedicated to the trial support, and also one to the communication, including patient and families empowerment. So this is just in a nutshell the project, so that you have the background information. But I think that the most important is to give the floor to the moderators and the speakers, and open the webinar session. So I give you the floor, Donato, for the introduction to the webinar. Thank you very much.
Thanks a lot, Christine. So I'm very happy to introduce this first webinar and first speakers. There will be a series of 3 webinars, April, May, and June, so very close one to the other. This is the first one, and we are going to present 2 tools that were developed within the PedCRIN project. The first one is on biosamples management. The second one is on pharmacovigilance, of course, both tailored to pediatric clinical trials with the very challenging issues we have to cope with during them. And the first speaker, I was leading together with Evelyn Jacques-Gren this work package 3, aimed to provide useful tools for pediatric and neonatal clinical trials. So this today webinar is dedicated to the pediatric tools, and I'm moderating it together with Evelyn from INSERM. The next one will be dedicated to the neonatal clinical trials tools. So we have our first speaker. She is Viviana Giannuzzi from Gianni Benzi Pharmacological Research Foundation, that is also a member of the PEDI Network of Excellence for Pediatric Research. So she's a long-lasting experience in mainly in ethic and regulatory issues that are the one more challenging within the management of pediatric biosamples. And she's currently also a member of the Pediatric Committee at the European Medicine Agency. So Viviana, please, the floor is yours.
Today, I will show you one of the tools developed in the context of the PedCRIN project. And this is on the biosample handling in pediatric clinical trials. So in this presentation, we will see briefly why do we need to manage biosamples in pediatric study, what kind of information need to be obtained for biosample management, and how is this PedCRIN tool useful, and at which stage of the trial it should be used. So let's start with very general concepts related with clinical trials that are used to include the use and storage of cells and tissues. Of course, they are related with the processing of health personal data. Sometimes we also need to transfer them. And in order to properly have all these activities related with the clinical trial as a whole, but also specifically for the properly use and storage of cell and tissue, we need to have an ethics approval and the authorization from the competent authorities. Sometimes the cell and tissues that are collected within clinical trials may be used for genetic tests. In this case, of course, we need to implement specific procedure in order to properly deal with this sensitive issue. If we look at specifically at the pediatric clinical trials, we know that they are characterized by well-known challenges, mainly related with the small sample size, the need for implementing appropriate procedures. For example, we needed to implement specific biomarkers on procedures because sometimes children, especially for the youngest one, are unable to communicate their feeling, the adverse events, and so on. We need to deal with a vulnerable population that is unable to provide its own consent. We have to minimize pain, discomfort, fear, and distress, and also we have regulatory burden because we have further regulatory requirement. Very often they are multinational studies, and families are not familiar with trials. They are worried about the risk of new treatment and so on. Importantly, we are dealing with a population made up of 5 different subpopulations, from the preterm newborn to the adolescents. For this reason, in the context of the PedCRIN project, an easy-to-use tool has been developed to guide investigators, sponsors, and other people involved in the pediatric trial to properly manage biosamples and associated data in fully compliance with the applicable European rules that are the standard that we need to comply with. What we did so far, we have identified the key topics and research questions. We searched and analyzed the applicable European provisions, and then identified the regulatory items, measure, and procedure have been divided in 5 main areas: consent and assent, minimizing harm and maximizing welfare, sampling volume, skills, training, and facilities required for sampling, and long-term storage of biological material. Let's start with the first topic, the informed consent and assent. All of us know that the consent is needed to enroll subjects in clinical trials. This process is not limited to the very beginning for the enrollment of the subject, but we need to be sure that the information provided to the subjects has been well comprehended. Then, of course, this is a voluntary decision, and then we need to be sure that the consent is maintained for the whole duration of the trial, even in case of reconsenting. In the case of pediatric clinical trials, of course, the subject is not allowed to consent by itself to participate in the trial because it's a minor. For this reason, we needed the consent from the parents and legal representative according to the national laws. So let's start with the first topic of our tool, consent and assent. And in our checklist, we listed all information that needed to be detailed in the information sheet and consent form for parents and legal representative related with use of biosample, of the handling of biosample in the information consent documents. So in the information sheet, we need to specify how biological material will be handled and used in the clinical trial, including the future uses. So we need to state which are the purposes of the processing of sample, as well as the future ones, of course, where applicable, the condition applicable to the storage, the condition governing the use of sample, of course, where applicable, the transfer policy according to the local and national laws, and the right to refuse consent and withdraw it at any time. Because, of course, this has an impact on the handling of biosamples. If genetic tests are foreseen in the trial, then we need to specify that, because we need to provide specific information in this case, and also a separate consent for genetic test. We need also to have specific information and to have a separate consent to process personal data according to the GDPR, that require us to specify in the information document a list of specific information related to the processing of personal data. Even in this case, let's think about the processing of personal data are linked to the biosample management. Here there are the long list of information that are required for data protection. So we need information about the data controller, the recipient, the data protection officer, the plan, the identification measure, and the period of storage of data. Even in this case, the initial and future purposes of processing. We still need to specify the applicable safeguard and the rights of the subjects. And where applicable, if there are the guarantee that are put in place for transferring personal data outside Europe. So what about the measure for destruction of sample and data in case the consent is withdrawn? We need to specify what has been put in place. In this case, the operation that are going to be performed in the trial must be in line with the consent. So where identifiable biological samples are stored for future purposes, for example, then the subject has the right to require that samples will be destroyed or anonymized in case of consent is withdrawn. Considering that in pediatric trials we have children to be enrolled, we need also to provide them adequate information according to the age and level of maturity. And we also need to collect from them their assent or agreement. So even in this, for information document for children, we need to specify what will happen to the sample taken. And even in this case, we need to specify information on genetic test. How to handle biological material already obtained when the subject reaches the age of legal competence to consent. This is also another issue that is related with a pediatric clinical trial. When the subject reaches the age of legal competence, the consent could be confirmed, modified, or withdrawn. We have specific information about that in the new clinical trial regulation. So if the subject does not take any action once becoming an adult, the consent given to the parent remains valid. If the subject withdraws the consent, sample and associated data must be destroyed or anonymized. So in your checklist, you can tick if all of these requirements are met in your trial or not. So you can adjust it, or they are not applicable. Another topic relates the specific procedure for pediatric trials. As I mentioned at the beginning, pain, discomfort, fear, distress, and any other foreseeable risk must be minimized. What to do? We need to limit study-related procedure to the most necessary, avoiding repeated invasive procedure, and we need to minimize the volume of blood withdrawing. So the second part of the checklist is related with that. You can tick if you have been put in place procedure to reduce painful procedure, for example, using microsampling, so using appropriate and age-appropriate assay material and devices, such as the needles, such as other validated procedure. We need to coordinate the timing of sampling to avoid repeated sampling. You need to treat possibly the physical pain and discomfort intensity according to the proper guideline. And you can also put in place methods such as patient population approaches and sparse sampling to reduce the number of blood samples in each child. Still continuing with the critical procedure to get biological material from pediatric patients. We need, as I mentioned, to minimize the painful procedure. Risk threshold, degree of distress, and physical pain must be defined in the protocol, so starting from the beginning, from the design of the protocol, and then constantly monitored during the whole conduct of the trial. As I mentioned before, the treatment should be effectively treated, and it should be reviewed regularly. We need to measure the level of the pain through ad hoc questionnaires, through the pain diary or self-report measures. We have a validated scale to measure the level of pain. And this is especially very important, considering that sometimes children, especially because of their age and their condition, are not able to express the level and intensity of the pain. Let's jump to the topic 3 related to the sampling of volume. We have a very specific, very useful recommendation in the European Commission recommendation updated in 2017. That suggests us which are the maximum volume of blood to be withdrawn from children, and we have information about the blood volume at any single time or over a period of 4 weeks. So we need to stay within these limits in our pediatric clinical trial. If applicable, we need to implement blood microsampling technique. in order to limit the blood withdrawn. We need to use low sample volume, we need to use micro methods on dry spot, and so on. Of course, even in this case, we need to implement specific procedure for children. And this should be, and if we cannot implement this technique, we should justify that in the protocol. In order to properly collect biosample for children, we need adequate facilities, and we need also trained professional to do that. So the fourth part of the checklist relates with that. We need to have professional expertise, qualified expertise with demonstrated experience on that, including and in particular for venipuncture. The facility need to respond to high quality standard and high quality requirement. We need appropriate facilities, for example, to minimize discomfort, fear and pain of children. We need a familiar environment and inpatient area to withdraw blood samples from children. As I mentioned, we of course need high quality standards for the facilities. We have here some of the requirement of the quality standard to be addressed. For example, those that BBMRI released assessment checklist on that. So we have standards to be met, and they are very clear and they're available as well. The last part of the checklist relates with the long-term storage of material. We need to foresee ad hoc measures to store the collected biosample, and the duration of the storage should be limited, and of course should be, as I mentioned, included in the informed consent documents, and the storage should be no longer than necessary and in compliance with the consent. Another topic is the future uses of biosamples collecting within a clinical trial. In fact, we can we can store part of the samples collected within a clinical trial for future secondary research uses. Of course, we need to implement a specific procedure in that case if we want to store and reuse samples of collected data. Also, the storage should be performed in a structured manner, and specific information should be provided in the information sheet with regards to the secondary use of samples and related data. Of course, the benefits coming from the research purposes from the secondary uses could be both of benefit for the patients, but also for other people, for example, affected by the same condition or the same category. This is to be specified in the informed consent and should be very well stated and justified. As I mentioned, we may need to transfer biosample data, for example, to another laboratory. This is particularly relevant and important in case of transfer across boundaries. So we need to follow specific rules in that case. Of course, we have no specific requirement for children, but we need to comply with rules applying for all clinical trials. So appropriate safety and confidentiality conditions should be done in accordance with the original consent given by the patients. We need to implement the identification measures according to the applicable framework, both at European and national level. We should transport samples not together with the associated data. In order to ensure that it is done according to an appropriate level of protection that should be ensured in all countries where samples are shared, we need to set up ad hoc data and material transfer agreement. So in our checklist, we can check if everything is has been properly set up in our clinical trial. This checklist is publicly available on the PedCRIN website, on the ACKNOWLEDGMENTS website. We hope it is an easy-to-use tool, and as I mentioned, it is to be used since from the design, the beginning of the design of the clinical trial. So while setting up the protocol, the clinical trial protocol, and it should be followed also during the whole conduct of the trial. So concluding what to do. What you need to do to properly collect, manage, and analyze and store biosample in pediatric trial. You need to follow the applicable ethical and regulatory requirement. Think about how to manage samples and associated data while designing the trial and then while conducting it. Include specific information on samples handling in the information sheet and consent form, as well as in documents for children. Implement recognized procedure to minimize pain. Foresee qualified facilities and expertise for personnel collecting biological material. Thank you.
Thanks a lot, Viviana. A very good presentation. Now we are going with the second presentation, and after it we will collect all the questions. So some of them will be related to the biosample management. So the second speaker is Cristina Manfredi. She is working at CVBF since 2005. So together with the start of the TEDdy FP6 founded project, and she's now the head of regulatory and pharmacovigilance area at CVBF. So please, Cristina, the floor is yours.
Thank you, Donato. Thank you also the organizers, Christine Salma and the ACKNOWLEDGMENTS team. Good afternoon, everyone. I know that there are a lot of people here today connected, and I would like to thank you all for your interest in our work. This afternoon, I will introduce you to a tool that can be used for the causality assessment of adverse events occurring during pediatric clinical trials. The evaluation of causality is one element of pharmacovigilance, Pharmacovigilance has been defined by the World Health Organization as the science and the related activities for detecting, assessing, understanding, and preventing adverse effects or any other medicine-related problem. This applies to all medicinal products for human use, including generics, herbal or complementary medicines, blood products, vaccines. The aim of pharmacovigilance is to improve public health, patient care, and safety in relation to the use of medicines, to contribute to the assessment of benefit, harm, effectiveness, and risk of medicines, and to promote understanding, education, and clinical training in pharmacovigilance and its effective communication to the public. Pharmacovigilance is even more important when we consider the pediatric population. Every child is a work in progress, starting from birth, and even before that, if we think about the intrauterine life, and up to the adolescence, important changes occur to children. Organs and systems continue to grow and mature. And as a consequence, children's bodies react differently from the adults, and disease and disorders may be different from their adult equivalents. This means that safety data cannot be necessarily extrapolated from data in adults. Children, so, may be more susceptible to ADRs from specific experience, or different ADR, which are adverse drug reaction, may be relevant for different pediatric age groups. Children may be susceptible to permanent effects that may result from a drug exposure at a sensitive point in their development. Pharmacovigilance is an iterative process. The detection that aims at detecting the unidentified safety issues, identifying risk factors, quantifying risks, and preventing patients from being adversely affected. As required by applicable laws, safety reports, and in case of pre-market pharmacovigilance, we are speaking about suspected unexpected serious adverse reactions, have to be submitted to regulatory authorities, ethics committees, and notified to sponsors. And in addition, we have annual safety reports that analyze serious adverse events, new safety findings, and evaluate the risk-benefit profile of the IMP, have to be produced during the course of a clinical trial. To detect unidentified safety issues, all adverse events have to be evaluated in terms of seriousness, causality, severity, and expectedness. Seriousness is regulated by law, by the reference legislation, and is based on the outcome of the event. The evaluation of seriousness is the first clinical assessment to be made and triggers the reporting to sponsors and regulatory authorities. Causality, instead, refers to a relationship between the drug and the adverse event. A correct causality assessment leads to better evaluation of the risk-benefit profile of the drugs and to signal detection. There are three different approaches that can be used to assess causality. We have the unstructured approach, which is based on medical experience and knowledge of the investigator or the assessor. The semi-structured approach, which provides a description of what is seen or not seen during an event and assigns a causality level. An example of this approach is the WHO UMC system for standardized case causality assessment. And finally, we have the standardized approach, which consists in the use of tools such as algorithms that have a set of question and decision rules, which leads the assessor to the final assessment. The Naranjo algorithm is the most well-known example of standardized approach for causality assessment. Severity refers to the intensity of an adverse event. We can have, for example, a severe headache. but it's not a synonymous of seriousness. It's based usually on the investigator's clinical judgment. And finally, we have expectedness, which is the second critical assessment that triggers reporting to regulatory authorities. It refers to events that have been previously observed and documented. Usually, when we think about we are dealing with clinical trials, the reference safety information are either the IB, the investigator's brochure, or the summary of product characteristics. Within the PedCRIN project, we focused our attentions on causality and severity. And we performed a systematic review focused on pediatric clinical studies using tools for causality and/or severity assessment. The period taken into consideration was between 1966 and 2017, and we retrieved a total of 151 studies that reported the use of tools. As you can see, in the figure, which is the results of our review, the 23% of the studies used the Naranjo algorithm, 8% reported to use the WHO system, and almost half of them did not specify any type of semi-structured or structured approach to causality assessment. The remaining 27% utilized different various methods and approaches. With reference with severity, the systematic review did not provide any useful results, and we extended the research using only two keywords, ADR and severity. This way we retrieved 149 publications, of which 14 mentioned the use of a tool. The Hartig and Siegel severity scales resulted to be the most used one. But our review led us to identify a new causality assessment tool, the Liverpool Adverse Drug Reaction Causality Assessment Tool, which was developed by the University of Liverpool in the framework of the Adverse Drug Reaction in Children Research Program, a project funded by the National Institute of Health Research. This causality assessment tool is a flowchart that was specifically adapted to the pediatric population on the basis of the Naranjo scale. The researchers of the PedCRIN program found difficulties in applying the Naranjo scales to events occurred with pediatric patients because, first of all, Naranjo is not a tool specifically thought for pediatric patients. They found it open to interpretation. It had found it to have some subjectivity, and that some questions were not appropriate for the pediatric population. And they found it difficult to create rules for individual questions with a heterogeneous case mix. And this is why they decided to develop an easy-to-use tool, while also ensuring that the basic principle of causality assessment were maintained. The results of their exercise was the LCAT, a flowchart that consists in 10 dichotomous questions that determine the path to the next question in an ordered sequence, eventually leading to a causality assessment. The first question of the LCAT is, do you suspect an adverse drug reaction? The definition of adverse drug reaction is the one that you find in the reference legislation, which is, for example, Article 2 of Directive 2001/20. And it is all untoward and unintended responses to an investigational medicinal product related to any dose administered. If the definition implies that there is a possibility of causal relationship between the event and the IMP. If you think that there is evidence or there are arguments that suggest a causal relationship, then your answer is yes, and you proceed with the LCAT to question number 2. If on the other hand, after looking at the evidence and at the information available, the answer is no, then it is unlikely that the event is an adverse drug reaction. Question 2, did the event appear after the drug was administered or dose increased? In this case, the assessor has to consider the temporal relationship between the onset of the event and the administration of the IMP. The relationship must be plausible, and the assessment should also take into consideration the pharmacokinetic properties and the half-life of the IMP. If the answer is no, you go to question 3. If the answer is yes, you go to question 4. Question 3 relates to the pre-existing symptoms and if they were exacerbated by the drug. And in this case, the assessor has to consider the symptoms that were already present before the IMP was administered, and they worsened after the IMP was taken by the patient. If the answer is no, then it's unlikely that the event is causally related to the IMP. If the answer is yes, the assessment proceeds with question number 4. So did the event improve when the drug was stopped or the dose reduced? The question evaluates information on the challenge of the IMP. And what you should think about is if the event improved after the IMP was stopped, and if it was, or if you cannot assess this because you have insufficient information or contradictory information, then you proceed to question 6. Otherwise, if your answer is no, you proceed to question 5. Where you evaluate if the event was associated with long-lasting disability or impairment. Disability is defined as any restriction or lack of the ability to perform an activity, or impairment is defined as loss of abnormal or abnormality of physiological, psychological, and anatomical structures and function. If considering this definition, your answer is yes, you proceed to question 6. If it's no, then the event might be possibly related to the IMP. Question 6 relates to the probability that the event was due to an underlying disease. The medical history of the patient and his or her current medical status should be considered for this question. And if your answer is high, you are high or you are unsure, you proceed to question 7. If your answer is low, you proceed to question 8. So, is there any objective evidence supportive of the causal ADR mechanism? By objective evidence, we mean things such as positive laboratory investigation of causal ADR mechanism, good evidence of dose-dependent relationship. And if you think that yes, there is objective evidence, you proceed to question 9. If you don't think that there is evidence, then you have a possible relationship of the IMP with the adverse events. Question 9 relates to the rechallenge and the positive rechallenge. The question evaluates information that responds to the question, did the event reappear after drug reintroduction? If your answer is yes, then you have a definitive causal relationship with the IMP. While if your answer is no, you proceed to question 9. And here in question 9, you are asked to evaluate if there is a past history of the same event with this drug in this patient. And in this case, you need to verify if the patient has had a similar reaction to a medicinal product belonging to the same class of drugs during a previous exposure. If your answer is no, then you proceed to the last question, question number 10. If your answer is yes, then again, you have a definite causal relationship with the IMP. And finally, the last question is about previously reported events with this drug. If you, and you need to consult the reference safety information of the IMP, so the IB or the SMPC, to answer the question. And if you look at it and you see that there are no previously reported events, such as the one that you have assessed, then you have a possible relationship with the IMP. While if you can see that there are being previously reported events related to this drug, with the use of this drug, you have a probable causality assessment. So the event is probably, IMP is probably the cause of the event. At the end, so you have, in summary, we are, depending on the final assessment you reach, you have either adverse events and serious adverse events with no reasonable possibility of relatedness with the study medication, and you have adverse drug reactions and serious adverse reactions that have reasonable possibility of relatedness with study medication. We, together with the LCAT tool, we have also developed a user guide with some practical examples that can help you with the use of this tool. And in addition, the University of Liverpool has prepared an e-learning package that can you reach at the websites that you find in this slide. I would like to thank Dr. Louise Bracken, Professor Matthew Peake, and Professor Mark Turner for who have collaborated with us for the development of the user guide and for making the e-learning package available, and Dr. Gallagher and the ADRIC Senior Investigation for the development of the LCAT. And this is everything from my side.